everyone, welcome to the chemistry sessions. So I hope you all are doing good and are the studies going great? I hope it is going super fine. So what we had started off in the last session was chemistry in everyday life, how I'm dealing with all the mechanisms and how I'm correlating all those chemicals that we have studied so far into our real lives. So that is basically what we are going to study in this very chapter, that is chemistry in everyday life. I hope you all have observed so many things around you which are nothing but the chemicals, from the air we breathe and from the clothes we wear, from the food we eat, from the medicines we take. Anything, be it anything, it would be nothing but related to what? Related to the elements of the periodic table, which together combine to form so many different kinds of chemicals. Researches are going on in each and every field in order to find out the chemical compositions and in order to find out new compounds which are going to make our lives easier, which are going to treat our conditions, our medical health problems, which are actually be treating as what kind of supplements in the food which makes our body healthier. So all those researches are still going on. So the people who are interested in knowing what all are the chemical formulas, chemical structures involved within the things around us, so you can also Google it, you can refer to so many books and you will find a lot of information about even the clothes that we wear. The dyes that we use are nothing but the chemicals. Maybe a resonance shift, maybe a resonance involvement of electrons can change the color of the dye even. So that is the beauty of the chemistry which is actually around us and which is surrounding us, or we are surrounding the chemistry, it's vice versa. So I hope you all have revised what we have done in the last session. We were actually focusing upon the medicines, the pharmacological effects of the chemicals, the medicinal effect of the chemicals. How can we use those chemicals? How can we incorporate those chemicals in our lives in order to treat the medical conditions? So that was what we were starting off. So in that case, we had discussed about the classification of drugs. So the medicinal chemicals are nothing but the drugs. Those drugs can be classified into different categories on the basis of their chemical structure, on the basis of their molecular targets, on the basis of what not. So basically these type of classifications is actually helping the doctors very much in order to assign a proper medicine to a patient. So that is how we are actually living our lives. So very interesting part we are going to discuss today is how the drug is actually acting on to its target in order to actually manipulate that target, or in order to inhibit that target, or in order to change that target so that the disease can be treated. So for that we will be discussing what are enzymes, what are receptors. I had given you the idea in the last session. So what is enzyme basically? Enzymes, the proteins, the biomolecules which are actually acting as the biological catalysts, making all the processes in our body. All the chemical processes involve the enzymes. These are the proteins which make our work easier. So basically under the mild conditions, it actually makes a chemical process go on. So that is how we are living our lives. So enzymes are actually the proteins and the biological catalysts. Also what we had discussed in the last class was about the receptors. What are the receptors? Again what? Again the proteins. So these are also the proteins. They are actually serving as the communication. They are providing the communication between the signals and the cells. So these are, they provide communication, serving as a bridge between the signals which are need to be transmitted within our body and what else, between the main cell to which the information has to be provided. So, between the signals and the cell. So, that is how actually the receptors work. These are nothing but the receivers of information. So, these are receivers of information. So, I hope that idea is very well clear to you. Let's move on to how actually these are the chemicals which are present in our body, and now I would like to discuss how I can make a drug act on these two enzymes, how the drug can be actually acting on these two enzymes which are serving as targets for that drugs. So, these are nothing but the targets present in our body, and I need one drug to supply to these targets, to make the drug attack onto these targets. So, that is what I am going to discuss today. So, here it would be known as the drug target interaction. So, that is drug target interaction. So, basically what kind of interaction would that be? For that purpose, first I would like to discuss for the enzymes. So, first is enzyme serving as a target. For drug. Now, here enzyme is acting as the target. So, first is that case. In order to discuss this very case, first I need to know what is the actual activity of an enzyme. So, here what is the work of enzyme? Work of enzyme, or I can say the type of catalytic activity. So, that is the question over here. So, what actually the enzyme does within our body, how it actually makes a process very easier to go on, how actually it catalyzes a chemical process to make it a simpler one, to decrease its activation energy. So, first thing what enzyme does is it provides an active site. If there is an enzyme, it has got a proper active site in which the substrate, what is the substrate? The reagent, or I can say the reactant which is need to be converted into a product. So, you must know that enzyme is nothing but a kind of a reagent which is acting, or I can say a kind of a catalyst. Catalyst is nothing but a reagent what we have taken in order to convert a reactant into a product. So, I have got a reactant, I need to make a new product. For that I would like to use the catalyst, that is the enzyme, which is making our work easier. So, this is the enzyme. Reactant can also be known as the substrate. So, what enzyme does is, what is the function of enzyme is, it provides active site to this very substrate to come and attach to it. So, it makes the substrate attached to it. So, the enzyme provides what kind of a site? Active site, provides an active site. What kind of an active site is it? It is a kind of a lock and a key model. You must have done this in your chapter surface chemistry. So, this is lock and key model. That means what is the lock and what is the key? The lock is the enzyme, the key is the substrate which is going to fit in. So basically the enzyme is serving as the lock, whereas the substrate is acting as a key. So this is known as lock and key model. Enzyme has such a hollow site, such a kind of a site built in, which actually attracts the substrate onto it and makes it gets attached. So enzyme provides an active site, not the site but site for substrate to get attached. That means how I am going to draw this very case. So here if you imagine that this is a something kind of a, let us just say this. So that is an enzyme if you say over here. So this is, I am taking as the enzyme. And I have got a substrate of this very shape. That is nothing but a substrate. So that is a substrate over here. So this substrate has a shape which can easily fit in over here. So that means this enzyme is providing a favorable site, an active site onto which the substrate can get attached. So here the attachment would be done something like this. So this is known as nothing but an active site, that is active site, or I can say a favorable site. And now this very things get attached. How does they get attached is something like this. Let me just draw that properly in order to show you. So that is how the two attachments will be. It is now trying to get in, it is now trying to fit in. This is the substrate here. So that is how it is going to fit in, like that. So that is how the substrate is coming on to the active site. So I hope that is very well clear to you. Now what happens here is one more thing which actually the enzyme does is, this was one of the things, providing an active site. Second thing is, enzyme provides certain functional groups which can attract certain functional groups which can attack the substrate very easily, which can attract the substrate easily. Also, this very kind of an interaction between the substrate and the enzyme is actually via maybe H bonding, it may be van der Waals forces, it may be covalent, and so on. So these are the kind of bondings which can actually happen in this case, or I can say even the ionic bonds. So anything could happen over here, any kind of an interaction will take place. So the other activity of the enzyme is, first what we have studied is the enzyme would provide an active site. What kind of an active site? A favorable site, such that the substrate can fit in very easily. So over here the substrate is getting fit very easily, and what do we have? A structure like this. This will now act and will give the new product. Also, what is the other function? It provides again the favorable functional groups attached onto the enzyme, which will attract the substrate towards itself. So that is also the another kind of function. So basically now we can, once we have judged the activity of the enzyme, now we can actually develop a particular kind of a drug which can inhibit its activity. So that is our main motive, that is our main target. So here now how does the drug will act? So now it comes the drug action. How it will act onto the enzyme? I am talking about the drug action on enzyme. By inhibiting, by preventing any of these processes, the drug will act. So it will, by inhibiting, or I can say preventing, any of these processes. So how does this actually inhibits or prevent these very processes? There must be some way out in order to actually stop these processes which are going on. So what is the first thing that the drug does? So here let's discuss the first thing which actually the drug will do. So the first thing is, it can actually mimic the shape of the substrate. So here what it can do is, number one, drug can mimic the shape of substrate. That means, what is mimic? Mimic is actually behaving of that sort. I am actually able to do the mimicry of Shah Rukh Khan. So basically what is mimicry? What is mimic? That means you are trying to act like that person. So here the drug is trying to act like that substrate. It is developing its shape in such a manner that it is actually replacing the substrate. So what will happen? The substrate, instead of getting attached to the enzyme, what will happen? The drug instead will get attached. So here what happens? It mimics, the drug can mimic the shape of the substrate. That means if I have got here, again I have got the enzyme over here. So here I have got the enzyme, and what about the substrate here? So the substrate looked like something this. So that is the substrate which we have got. And what about the drug? So the drug will also be having a similar kind of a shape. So that is, again, this is the enzyme that we have taken initially, and that is the substrate. So here it's enzyme and here it's the substrate. What will it do is, now here comes the drug, our main warrior, our main fighter. So the drug will be like this. It is actually behaving as, so that is our drug over here, having a similar kind of a shape as that of the substrate. It will now replace the position of the substrate from the enzyme. So this is nothing but the drug. having similar shape. So instead of this getting attached over here, this will not. Instead, the drug will come and get attached over here. So that is how the drug is actually creating a sort of confusion between the enzyme and the substrate. The substrate will not actually now get attached, but instead the drug. So what will happen here? We get something like this. So much of interesting kind of a work this very drug is doing. And what do we have here? Attachment of the drug here. So that is how it is inhibiting the activity of this substrate actually. So here I have, this is nothing but the enzyme over here. So that is the enzyme and that is the drug. So here what it has does is, drug here has inhibited the substrate enzyme interaction. So that is how the drug is acting. So I hope that is very well clear to you, and I hope you all have actually observed what kind of an interaction is taking place between the drug and the enzyme this time, not the substrate and the enzyme. So that is how it is actually preventing such kind of an activity over here, and it itself gets attached over here. So I hope that is clear. Now let's move on to the second thing. What does the drug do? So here is the second thing that we are going to discuss. So basically the second thing is, whenever there is the substrate, I have got the substrate here. So that is how the substrate actually looks like. Again, so basically the drug does not gets fit in over here, but it actually fits in other kind of different kind of a site which is present in the enzyme. So that different kind of a site present in the enzyme is maybe something like this. I am just drawing it randomly. So that is the another kind of site. This is known as allosteric site. It's a different site. Here what happens, the drug gets attached. So onto this site, on this site, drug gets attached. So whenever the drug gets attached over here, it changes the shape of the active site. So here after attachment, so let me just have an attachment over here. I just need to show you. So maybe there is the drug which is getting attached. That is the drug. I hope that is clearly visible to you. Drug attached here. What it does is, it changes the main active site over here. So it change of active site of shape of active site. Now what will happen? What will happen is, we obtain a structure like this. The shape has now changed and the drug is still attached. So I have got the drug over here. So that is the drug attached. And what else do I need? Now what will happen? If I have got a substrate over here, a substrate like this, this was the kind of substrate that we had in the initial, at the very beginning. So here what happened was, so I got, this was the initial substrate, and I cannot let this go in. That is not possible. So here what will happen? The substrate got confused and it is not able to fit in properly. So here the shape is changed shape, and what is happening? Substrate cannot enter. So that is what actually is happening. This is another kind of an action of drug. First, the drug actually attaches itself onto an allosteric site. So it got attached, and due to this attachment, what happens? There is the change of the shape of the main active site. So the shape changes, and now the substrate cannot enter into that active site, inhibiting the action, inhibiting the interaction between the enzyme and the substrate. So I hope that is very well clear to you. So here what happened was, drug changed the shape of active site, making the substrate enzyme interaction impossible, thereby inhibiting the enzyme action. So such evil is the drug. It actually, what does is, it can either mimic the shape of the initial substrate and itself gets attached, or what it can does is, it actually binds onto some of the allosteric sites and it can change the shape of the active site even. That is thereby inhibiting the enzyme action. So due to this kind of an interaction, due to this kind of an action, what happens is, the enzyme drug, the complex which has been formed now. So here the enzyme drug complex formed is then useless, and is discarded by the body. Thus new enzyme is produced. New enzyme is released, new enzyme is produced. So that is what is actually the drug action. How evil the drug can be belonging to its target. So if the drug has got the target, by hook or by crook, it will change the activity of the enzyme. It will change the activity of enzyme and substrate interaction, inhibiting the chemical process. Now the chemical process is very, very difficult to happen. So that is how the drug acts. I hope that is very well clear to you. So just note this down, then I will be taking up the drug receptor interaction. So I hope you all have noted this down. Now let's move on to how actually the receptor, its communication is how broken by the drug. So let us just see how. I am rubbing this off. So how does the receptors work? Actually, they are actually acting as the receivers of information. So that is how they are working. They also have got a site onto them, just like what we have in the case of enzyme. So the receptors are present receptor drug interaction. So what is happening is receptors are firstly are present. They are present in the cell membrane. So the outer as well as the inner cell membrane. So it is covering the both. So receptors are present in cell membranes. So how can I show you that the receptors are embedded in such a manner into the cell membrane? So see, if I've got this as a receptor, so this, if I make this as the receptor over here, so that is a receptor. Let's just say this is the kind of a shape of the receptor, acting as a receiver of the signals within the body. So that is what we have in the case of receptor, and this is the binding site. And what do I have here? I have got this as the outer cell membrane, and these vary as the inner cell membrane. Again the outer cell membrane, and the inner cell membrane. So that is how actually the cells are, the membranes are arranged, and that is how the receptor has been binded. So this is a receptor. These are the remaining membranes over here, the layers. Now that is how the receptor is, and this is the outer cell membrane. And that is the inner cell membrane. So the binding site, let me just write it over here. The binding site, or I can say the receiver site, is projecting outwards from the outer cell membrane, Just mind it, I am talking about the animal cell membrane over here. This is nothing but animal cell membrane. So now what happens? There is some kind of a site over here which will receive something. So what is that something over here? It is the chemical messenger. So basically what happens is, some kind of signals, some kind of information is passed on from one neuron to the another. So basically that neuron now transmits the information onto the muscles. So how does it take place? it is carried forward, that information from neuron to muscle is carried forward by the chemical messenger, and that chemical messenger is actually passing on the information first onto this receptor. So it gets fixed over here, and then this receptor will pass on the information to the cells, to the further cells in the body. It could be the muscle cells. So I hope that action is clear to you, how actually the signals are getting transmitted. So let me just write it over here. How signals or information is getting transmitted, in body. So basically I need to pass on the information. The information is going from one neuron to another. So information is passed on from one neuron to another. To another. Now the neuron will transmit the information to the cells. So let us just say neuron is going to transfer or going to transmit it to the muscle cells, the information I am talking about. So this information is passed through a chemical known as chemical messenger. So now how does this chemical messenger is actually acting? So what happens is, let me just first rub this off. I hope you all have noted this down. So what happens is, the chemical messenger binds or interacts at the active site of receptor. It binds or interacts with the active site of the receptor over here. The shape of the chemical messenger would be something like this only. So what will happen now? It will finally, the receptor will receive the information, it will finally transmit onto the cell. So here is the cell over here, it is going to transmit the information like such. So what happens is, the chemical messenger first binds, then the information is taken up by the receptor, and finally it is passed on to the cells. Onto the cells. So that is how basically what the main process is. Just like what we have did in the case of enzyme substrate interaction, that was the main functioning of enzyme and substrate interaction. Now this is the functioning of receptor, how the receptors are working within our body in order to transmit, in order to communicate the information. So basically what happens is, let me just repeat this off, the receptors are there just like that, embedded into the cell membrane, and the outer portion, which is the receiving side over here, is actually present towards the outer cell membrane, that is outside the cell. So what will happen is, when the information is carried out from neuron to the another and then neuron to the muscles. So that information from neuron to the muscle is transferred via the chemical messenger. A chemical which is a messenger, that means it carries the information. So this messenger comes on and it interacts onto the binding site. What will happen? It further transmits the information to the receptor, which is the receiver of information, and the receiver then transmits the whole information onto the cell, thereby making whole communication process feasible. So that is what functioning of receptor is. So in the next session I will be discussing of how does actually the drug is now acting as an evil and will inhibit all of these functions very cleverly, very confidently. How does the action takes place? So we will be discussing this in the next session. Till then, just revise what we have done so far. Have a good day.